Hello, we are students from Harmony School of Advancement, and our PBL is called Early Detection of Influenza. My name is Amy.
My name is Alan.
And my name is Destiny.
The goal of this project is to have pictures showing gel electrophoresis results. We seek to apply this data to lessen the impacts of yearly seasonal influenza epidemics during spring and winter times. We also seek to acknowledge the following question. If influenza is detected early on with RT-PCR tests, will they be impactful and contribute to fewer hospitalizations?
Historically, influenza was detected through ELISA tests. However, critics argue that ELISA tests take too long to perform. As a result, rapid influenza diagnostic tests, also known as RIDTs, were developed to solve this issue.
However, RIDTs often lead to inaccurate results, such as false positives and negatives, thus limiting the effectiveness of prevention. So scientists have utilized a procedure called reverse transcriptase polymerase chain reaction. RT-PCR works by using an enzyme called reverse transcriptase to create more copies of the complementary DNA of the influenza gene.
Once the reverse transcriptase finishes the synthesization process, the cDNA gets mixed with Taq polymerase, dNTPs, and primers for PCR amplification.
Once the amplification process is complete, a gel electrophoresis is run to determine the results. RT-PCR is often considered the gold standard of influenza detection because of its high specificity and sensitivity.
In our experiment, we followed the procedures set by Advotech and performed two tests, RIDT and a RT-PCR in a clinical stimulation.
Before our experiment, we had performed needed lab preparations. First, we had to prep the RIDT strips by applying one drop of simulated antibody solution onto each of the circles.
Then, after waiting a few minutes for the strip to completely dry, our results were shown. If the spot shows up pink, it is a positive result, meaning that the influenza antigens were detected. If there is no pink color, the result is negative.
With the RIDT test completed, we began the necessary lab preparations for the gel electrophoresis part of the RT-PCR test. In our experiment, the RT-PCR test was already completed for us by Advotech, and we were required to do the gel electrophoresis portion.
We first had to prepare the agarose gel by dissolving the agarose powder with distilled water. While the instructions say to dissolve the powder using a microwave, we utilized a hot plate to dissolve it. After it cooled to 60 degrees Celsius, we poured the solution into a gel mold, capped it. We then left it to solidify for 20 minutes.
After the gel solidified, we prepared the buffer solution by pouring the specific amount of diluted water and buffer concentration given by the guidelines.
Once the buffer solution was made, we slid the agarose gel with the tray still on into the gel electrophoresis chamber. Then we poured the diluted buffer solution into the chamber, making sure to submerge the gel completely.
Once again, we began pipetting the samples into the correct wells of the gel. Once all samples had been placed, we ran the gel electrophoresis for about 30 to 45 minutes.
After the gel electrophoresis had been completed, we began the staining process. We diluted the staining solution with distilled water and then poured the staining solution onto the gel.
After the staining process was completed, we destained the gel with warm water for about 10 minutes.
We then removed the excess water and began the viewing process under UV light. Shown below are our results. The first line depicts the DNA standard markers, the second line depicts the negative control, and the third line shows a positive control.
The last three wells feature patients 1, 2, and 3 respectively. By comparing the controls, patients 1 and 3 test negative for influenza while patient 2 tests positive. By comparing the RT-PCR gel electrophoresis results with the RIDT test strip, we can see that patient 3 was incorrectly misdiagnosed. This shows that RT-PCR's level of accuracy can be used to help correctly diagnose patients and thus slow the spread of influenza.
This was our PBL on the early detection of influenza, and we hope you learned much from it. Thank you.